Welcome to the Cytology Lab Practical. For the benefits of this particular exercise, I've taken the exercise down to the Cytology Lab in Derriford Hospital, who's kindly let me film exactly what happens in a real laboratory. So once the sample arrives, first of all, goes straight into the fume hood. This is of course because of potential for pathogenic infection, so you need a continual, appropriate stream of air and aseptic technique to ensure that, well, one, nothing happens to you, and two, nothing happens to contaminate the sample. Depending on what happens here, if it's a particularly mucoid one, such as from a lung, you may need to either spread it directly onto the slide, you can have alternatives for preparing it to break down mucus, or you can spin it down in a special piece of kit to put it on the slide as a thin film. So to do that, you have a couple of centrifuges, two different types here. This particular one here is for big samples, and this particular one uses a special piece of kit that will make a very pretty little slide at the very end. The likes of which gets done over here. This is the staining station, and the staining station is for this, is the Pap stain, Papa Nicolau, who's old George Papa Nicolau, who had nothing better to do during the Second World War than devise this little fella. And the end result will be a stain that is pretty much blue, but gives you a lot of information, all to do with the cytoplasmic detail. The Pap stain process follows a very simple, logical and sequential order, much the same way that the hematoxylin and eostin stain does that you have done previously. So to start, it begins off in 100% alcohol. That is a fixative and prevents any kind of infection spreading. Then into a 70% alcohol, into a tap water wash, hematoxylin for the nuclear detail, acid alcohol to remove the excess. Scott's solution is our cheat solution, if you remember, purely because Devon water is so soft that the hematoxylin doesn't blue very effectively. So Scott's cheat does that for you. Now here, unlike the hematoxylin and eosin, there's a slight difference. We put it into alcohol. And the reason for that is that OG6, orange green 6, is an alcohol-based dye. So you start from Scott's into alcohol to make sure you've got it completely into an alcoholic solution, into the orange green, which will stain keratin orange. Then you wash out the excess, which is why you've got these two different sort of yellowish colours. Obviously that's got more residual stain, that's got less residual stain. Straight into EA50. And this is a polymeric dye. This provides the sort of a greenish light blue colour. Once that's done, washing out in IDA again, IDA, xylene and xylene, because that will allow us to put a cover slip on. And the end result will be some of the photographs which we'll look at right in a minute.